So, good afternoon to everyone. Uh, we've joined for the Climb India Journal Club this month. This month, we're privileged to have our esteemed team from SIMS Nagpur, who will be presenting a very interesting topic on the blood-brain barrier. So, there are so much of research which actually goes on about blood-brain barrier, and uh, we all know that blood-brain barrier is very unique and extremely foolproof. We call the brain a privileged organ, but all the organisms, for some reason, are prove much more intelligent, than the existing brain, and they find a way through. But that also is a difficult task, and I'm happy to introduce Dr. Rajpal Singh Kashyap, who's the director of research at SIMS Nagpur. And Sir has done a lot of work, and his niche area of interest is in the blood-brain barrier and studying the biomarkers of inflammation. We're happy to have him as the moderator for this session. And I'm happy to introduce two bright young stars who are actually project technical support 3 and 2 in this Climb India project, which is all of you know, which is a Center for Advanced Research grant on studying the impact of climate change and brain infections. So we have Ms. Komal and Ms. Vaidehi. Both of them have actually are going to present, and this is the first of the kind where the institute's project technical support taking the lead in hosting this seminar. So I would like to thank the team because they've opened out a new avenue for the other teams to follow. So over to Sir for moderation.
Yeah, thank you, Dr. Angel. I think it's all because of you, because you have started this kind of a trend. And now I think today we have two important things. One is the topic, which you have already indicated that it's a very important topic. And the second thing is, I think we should give chance to our budding scientists because they should face this kind of platform which you have created, and definitely some good, some bad. They'll feel some experience, some right, some wrong, but actually they'll feel the heat, and then we see people like you as a role model, like you are getting this car project and doing a lot of research. So maybe after 5-10 years they'll come to your place, and so this is the thing. I personally feel that we all should work together, give chance to others. I mean, clinician can present. A lot of clinicians are here; they can present. But I think it's a time for me also to see what they have understand, right? Because it's a time because we have hired them for this project. So let's see what they project. Now, coming to the topic, before I hand over to our budding scientist for this journal club, BBB is really a difficult thing, and and when we talk about research, we do get some papers, we do get some ideas, but when we actually go to the bedside, it's very difficult to convert all those papers into real-time experience with the clinician and all these things. The results are different. So whether it is a transport of drug, I mean, basically the drugs goes to the brain, the limited drugs goes to the brain. This is the issue. And then we have how the bug crosses the blood-brain barrier. This is the issue. And ultimately, we don't know about all these things. And even we don't know about the how with climate change BBB work out. So I think there are several things. So what they have decided, they have talked to me once. So what they have decided to have 5 concept, 1, 2, 3, 4, 5, and have some paper to prove those 5 concept, so that a story should be continued, and some references should be there, and they'll come out with some kind of a research presentation as well as journal thing. So I think this is a new experiment, and let's see how the things will come out from these things. Questions, we'll understand more about all these things. They will understand more about these things. And next project, Dr. Angel, we have to write on a BBB, because that model has to be established in a cell culture, 3D culture, or something like that. And then we need to work on this project, and ICMR will definitely looking into it. I have presented several times in ICMR also. So let's see what we gain out of this presentation, which they are going to present. So I happy to invite our budding scientist Komal and Vaidehi, and please go ahead with your presentation. That's all, Angela, I have to do. Yeah, you are mute.
I'm sorry. Yeah.
Yeah.
So as they present, if you you can pitch in between if you want to give any expert comments and opinion.
Yeah, absolutely.
Otherwise, we could wait till they finish and then we can have a discussion with the audience. That
Wherever
is
it's
also
required, I'll just come into picture. That's
yes,
not a problem.
yes, that's fine. Okay,
Thank
sir.
you.
Komal,
Thank you,
you
sir.
can share your
Thank you, ma'am.
slides. Yeah.
Yeah, sure. Ma'am, is my screen visible?
Yes, yes. You can put it in slideshow mode, please.
Sure. As you said, the brain is the most protected organs in the body, yet some infections still manage to cross the line. Then I just wanted to say, have we ever wondered what determines whether an infection stays at the periphery or finds its way into the brain?
The answers lie in a remarkable structure, BBB, blood-brain barrier. It's not just a ball, it's a dynamic living interface. Today we will explore how it works, how pathogens break it, why the immune response often makes things worse, and what new tools are have to fix it.
So I am Komal.
I'm Vaidehi Motghare. Under the guidance of Dr. Rajpal Singh Kashyap, we will take you through 5 evidence-based sections. Thank you.
I think Vaidehi, your voice, you might have to probably come a bit closer to the screen because
Yeah, that's what I'm saying. Komal, once you finish, maybe you ask whether to come down to your place or whether they should come to the mic near to the mic.
Sure.
Because his
Sure,
voice is
sir.
not clear. Our voice is not clear.
yeah.
Okay, sir. So we have structured our talk into 5 evidence-based sections. First, the biology of the blood-brain barrier, how it functions as a door and its lock. Second section will be about pathogen-based blood-brain barrier injury. Third section will be about immune-mediated based neuroinflammation. And fourth will be biomarkers and prognosis. And fifth will be future therapies and blood-brain barrier models. The first 3 sections will be covered by me, and the section fourth and fifth will be covered by Betty. So let's start with the basic. Imagine a fortress protecting the most valuable asset you have, your brain. The blood brain barrier is that fortress. It's not a single wall, but a complex structure called the neurovascular unit. At the core, there are endothelial cells that line the blood vessels. Between these cells are the tight junction proteins like Claudin 5, occludin, zooxanthellae 1s, which act as heavy duty locks, the ceiling gaps basically. Wrapped around outside are the pericytes. Think of them as the security guards, and beyond them are the astrocytes and feed that send signals to maintain the barrier. There is also a basement membrane that provides the structural support. Now, researchers measure how tight is this blood-brain barrier using a tier technique, which is transendothelial electrical resistance. High tier means you have a tight barrier. Now, in healthy humans, this value is over 100 ohms per square centimeter, but as we'll see in the next slides, this door is not static and it's not completely impenetrable. So, if the BBB is so effective, why do some people still get brain infections? Let's look at the numbers, the clinical puzzle. Now, there is a clinical puzzle that motivated our entire talk. Look at these numbers. First, a 2009 study by Von Well et al., in which they stated that 87% of the children with TBM have no miliary TB on their chest radiographs. That means that a majority of the TBM patients never had widespread lung disease. Second, a 2020 review by Vasconcelos et al. Looked at patients with miliary TB where the bacteria have spread through the blood to many organs. They found that CNS involvement occurs in only 10 to 30% of the cases. So, when the bacteria are everywhere in the bloodstream, the barrier usually holds. What does this tell us? The disseminated TB does not guarantee brain infection, and TBM can happen without disseminated disease. The barrier is selective, breach is not automatic. It requires a specific bacterial strain, host genetics, and the right immune conditions. With that foundation, now let's see what what this clinical puzzle sets a stage for. So the clinical puzzle tells us the barrier failure is not just about the amount of bacteria, but the barrier itself is not a static valve. It actually changes over 24 hours. Let's look at the evidence for that. Building on that clinical puzzle, now we see that BBB is not just a static store, it has a daily rhythm called the circadian regulation. A 2025 review by Elliott et al., they show that a P glycoprotein, that is an efflux plump that kicks drugs and toxins out of the brain, and it peaks during the active phase, that is night for rodents and day for humans. In contrast, the tight junction protein claudin 5 peaks during the inactive phase, the opposite rhythm. Means the P glycoprotein and the claudin 5 works antagonistically with each other. These rhythms are driven by a molecular clock inside the endothelial cells themselves, like the clock and BML1. In fact, the 2018 paper by Zhang and colleagues also showed that the BBB has its own intrinsic clock which regulates the efflux. So what does this mean for the infection? Pathogen entry may be time dependent. If the barrier is more permeable at night, a bacterium that enters the blood at the night might have an easier time crossing. And conversely, if we want to deliver drugs to the brain, timing matters. The master clock that drives all of this rhythm is a tiny region in the brain called the suprachiasmatic nucleus, or the HCN, located in the hypothalamus. It synchronizes our body's daily rhythm, sleep-wake cycles, hormone release, and now we know the BBB permeability. So understanding this rhythm has a direct clinical application. If we know the barrier is most permeable, we can time the therapeutics and translate them more effectively. So how can we use this rhythm clinically? This idea is called the chronotherapy. Here is the key. P glycoprotein, the efflux pump that kicks drugs out of the brain, is slowest during the rest phase. In humans, that's at night. In rodents, it's during the day. Drugs that are PGP substrates, meaning they will get pumped out, they will get pumped out during the inactive phase, will penetrate the brain much better when given during the low PGP window. Elliott et al. In their 2025 review explicitly state, and I quote, by understanding how CNS barrier function fluctuates across the circadian day, additional chronotherapy approaches may be identified. Here is a concrete example by a 2018 Cell paper by Zhang et al. What they did was they studied an antileptic drug, phenytoin, which is a PGP substrate. When they gave it to the mice during the night, when the PGP was low, the brain levels were uh, significantly higher when they were given during the day. The same dose at the different time worked better. Now, this hasn't been tested for anti-TB drugs yet, but drugs like rifampicin are also PGP substrate. So giving them at night when PGP is low could potentially increase the brain penetration. So the BBB has a daily so the BBB has a daily rhythm that may affect the drug delivery. But the more fundamental question is, how do pathogens like Mycobacterium tuberculosis cross the barrier in the first place? And why do some people get TBM and while others do not, even with the similar exposure? So the BBB has a daily rhythm. We all saw that in the previous slide. That's an exciting area, but the more fundamental question lies the same. How do the Mycobacterium tuberculosis crosses the barrier in the first place? That brings us to the section 2, that is pathogen-induced blood-brain barrier injury. In this section, we'll see that the strain of the bacteria itself, not just immune system, determines whether you get a brain infection. We'll look at the three lines of evidence. First is the C3 model. First is the C3 mouse model by Dr. Uh, Rajpal Singh Kashyap, which shows that a CSF-derived strain enters crosses the blood-brain barrier and reaches the brain, while a sputum-derived strain did not. Now, this paper is the anchor of our entire talk. This is a 2017 paper published in the Indian Journal of Medical Research. So let me walk you through what exactly they did. They isolated a clinical strain of Mycobacterium tuberculosis from the cerebrospinal fluid of a patient with confirmed tuberculous meningitis. That patient had a normal X-ray, no evidence of lung TB. They call that strain C3 for CSF derived. For comparison, they took a sputum strain from a patient with only pulmonary tuberculosis and no brain involvement. They injected the strains intravenously into the healthy BBB mice, a high dose of 2 into 10 to the power 7 CFU to ensure consistent infection. They waited and measured what happened at 30 days and 50 days. At 30 days, the bacteria was mainly in the lungs, and the brain had a low but detectable load. But there is a critical observation: no inflammation markers, no ADA, no antibodies, no antigens were detectable at this time. But at day 50, something dramatic happened. The lung bacterial load had dropped, and the brain load was increased up to 10-fold, and now suddenly the brain ADA spiked. The antigens like anti-585b and RV2623 became detectable. So histopathology also revealed uh, some changes, which we'll see in the next slide. The survival Mayer graph also uh, show the striking data that 60% of the C3 infected mice dies by the end of the experiment. In contrast, the sputum strain given the same dose, same route, caused minimal brain infection, low mortality, and persistent lung pathology. So what does this mean? The bacteria moved from lung to brain over the time. Hematogenesis spread, but the key finding is that the strain's origin predicted the neuroinvasion. The CSF-derived C3 strain caused the blood-brain barrier, but the pulmonary strain did not. This is the direct. This directly explains the clinical puzzle that we saw earlier. That even with the miliary TB, where bacteria were everywhere in the blood, not every strain can cross. TBM requires a specific CNS adapted strain. Now, could it be that the C3 strain just happened to be more virulent in general? That's the next slide from Hernandez Pando et al. They used a more natural route of infection, intratracheal, directly into the lungs, and they tested three different strains from meningitis patients, strains 28, 136, and 209. For comparison, they used a pulmonary strain, including some virulent strains, including H37RV, Beijing family strains, and the Lep strains. What did they find? All the three CSF isolated strains caused the brain infections. Strain 209 was hypervirulent though. It killed the mice quickly and caused very high bacterial loads in lung, liver, spleen, and uh, blood. But even with the less virulent strains, 28 and 136 also crossed the brain barrier and infected the brain. In the striking contrast, the pulmonary strains, even those that were hypervirulent in the lungs, caused minimal brain infection. The same was true for H37RV. So here is the key takeaway. Neuroinvasiveness is a specific property of the CSF derived strain, not a general consequence of being virulent. You can have a pulmonary strain that is highly virulent in the lungs, like a Beijing strain, but it is still not able to cross the blood-brain barrier efficiently. This also explains the clinical puzzle from the slide 1.2. Why do many miliary TB patients never develop TBM? Because the strain that they carry may simply lack the genetic ability to develop TBM, because even if it's spreading everywhere, it's not able to cross the blood-brain barrier. So we have established that CSF strains have a special ability to cross the blood-brain barrier. What makes them neuroinvasive? Let's look into two factors: the bacterial genes and the host factors. On the pathogen side, a gene called PKS15-1 produces a molecule called phenolic glycolipid, or PGL. PGL suppresses the innate immune response. It basically tells the immune system to stand down, allowing the bacteria to spread beyond the lungs. This was shown by Sinsimur and colleagues in 2008 and B and colleagues in 2011. But here is the twist. Strains from Euro-American linkage, which include many of the strains circulating in India, lack an intact PKS15-1 gene. Yet some of them still cause TBM, as we saw with Dr. Kashyap's papers, C3 strain. So while PKS is important, it is not only factor. Other genes like Other genes may be overexpressed, which may include a lot of bioinformatics work to see how much is the overexpression or not. Now the host side, even if you are infected with a neuroinvasive strain, not everyone gets TBM. That's where the host genetics come in. Now the genetic studies from Vietnam and Horn et al. Identified single nucleotide polymorphisms (SNPs) into two immune receptor genes, TIRAP and TLR2. These SNPs were strongly associated with increased susceptibility to TBM even when the patients were infected with the same strain. TLR2 is a receptor on immune cell that recognizes components of TB bacterium, and TIRAP is an adaptive protein that helps transmit the signal from TLR2. If either of these genes has a variant that changes function, the immune response may be less effective containing the bacteria, allowing them to cross the BBB. So here is the bottom line. BBB breach is not purely pathogen driven. It's the collision of specific bacterial virulence factors like PGL and other unknown genes and host immune genetics that determines if the barrier falls. Let's summarize what we have saw in section 2. The table tells us the story simply. The CSF derived C3 strain, when injected intravenously, called high brain bacterial load, 5 log CFU, and 60% mortality. And notice the lung pathology. It actually improves over time. And in contrast, the sputum strain, same dose, same route, caused minimal infection in the brain. It had low mortality, and the lung pathology did not improve over time. It was same, and it showed persistent pathology. So now, once the bacteria crosses the blood-brain barrier, the real troubles start. Real troubles begins not necessarily from the bacteria themselves, but from the immune response they trigger. That's what we'll explore in section 3, the friendly fire problem. That is the immune-mediated neuroinflammation. Now we come to the paradox, as this is where the story gets really interesting. We are going back to Dr. Kashyap's 2070 C3 strain paper, but now we are looking at the timing of inflammation. Look at the table. At day 30, the bacteria are already in the brain, 4 log CFU, but here is the critical observation. The lungs are also heavily infected, 6.8 log. But here is the critical observation. There is no observation of inflammation, no ADA, no antigens, no antibodies, and BBB is still intact. When we look at day 50, the lung bacterial load has dropped and the brain load has significantly increased to 5 log, and more importantly ADA spikes dramatically. That's the marker of T cell activation, and on the histology sites you may see a massive lymphatic infiltration, immune cells pouring into the brain parenchyma. You also see a neuronal swelling and edema, clear signs of damage. So here is the paradox. The bacteria were already in the brain at day 30, but the damage did not really start until day 50 when the T cells arrived. Where the immune response, not the bacteria themselves, is what causes the BBB breakdown and the neuronal injury. That a critical insight. If you could block the T response without compromising the bacterial load, you might reduce the damage. But of course, that's a delicate balance. You still need to kill the bacteria. So, how do we measure this T cell driven neuroinflammation? That brings us to a ADA, a simple, beautiful biomarker. So, ADA is an enzyme produced by activated T cell lymphocytes. When T cells are activated, as they are in tuberculous meningitis, they release ADA into the surrounding tissue and the cerebrospinal paper. In the C3 mouse model paper, they measured ADA in the brain homogenetes of infected mice at day 30 when the bacteria was present, inflammation was low, ADA levels were barely detectable, but by day 50, the ADA levels spiked dramatically, and the difference from the uninfected control was highly significant. That is, p-value was less than 0.0001. Now, here is where this becomes clinically powerful. That CSF ADA levels above greater than 10 units per liter really distinguish tuberculous meningitis from other cases of meningitis like bacterial, viral, or fungal. The sensitivity and specificity were both over 90% in the study, and it is a simple, inexpensive, widely available technique. So, why does this work? In bacterial meningitis, the immune response is dominated by neutrophils, which don't produce much ADA. In viral meningitis, the T cell response is there but is often milder. In TBM, you get this intense chronic T cell activation and the ADA goes up. And here is the key point: elevated ADA is not just a diagnostic test, it's a direct surrogate for the neuroinflammatory response that is damaging the BBB. High ADA means active T cell attack on the brain, and that attack we saw, which drives the pathology. But which cells are actually doing the damage? Is it only the invading T cells, or are the brain's own immune cells? The microglia are also involved. That brings us to the most surprising part of the section 3. Before we move to the biomarkers, let's see what we have seen in TBM and what happens in viral encephalitis, because the final common pathway is the same. Both, but the triggers are different. Look at the table in bacterial meningitis. Specifically TBM as a model, the dominant cytokines are TNF alpha, IL-1 beta, IL-6, and interferon gamma. The BBB disruption is largely immune cell mediated. T cells infiltrate, microglia become phagocytic, and astrocyte end feet are destroyed. In viral encephalitis, like Japanese encephalitis or long COVID, the story is different. For JV, the viral NS1 and NS2 proteins directly activate the TLR4, leading to different driven autophagy and the Claudin 5 um, degradation. The dominant cytokines include IL-6 and type 1 interferon. For SARS-CoV, the S1 spike protein directly spikes that directly induces the TNF. So in long COVID patients with brain fog, DCEMI shows a clear BBB leakage and the cytokine TGF beta is specifically elevated. So the common pathway is the same. Pathogen will activate TLR, NF kappa beta, cytokine, then it will lead to cytokine storm, tight junction degradation, BBB failure, neurological damage, but the trigger, whether it's a live bacteria, viral proteins, or persistent spike protein, and the dominant cytokines differ. The broader perspective is important because it tells us that while we may need the pathogen specific diagnostics therapies that stabilize the blood brain barrier or modulate the host response could work across multiple neuroinfection. Now, with that understanding, let's see the next slide. The 2024 Nature Neuroscience paper by Green et al. Used dynamic contrast agent that normally does not cross the BBB. If the barrier is leaky, the contrast agent extravasates into the brain tissue and you see the bright areas on the scan. The top images are from the recovered COVID patients and brain fog, and you can see the bright areas indicating BBB leakage, specifically in the temporal and frontal lobes. The paper also found that the TGF beta, an anti inflammatory cytokine, was specifically elevated in the brain fog group. Uh, and they showed that S1 spike protein directly activates the brain endothelial cell. This is a powerful evidence that the BBB disruption is not just a mouse model phenomena, it happens in humans. It's measurable and it correlates with the real neurological symptoms. Now, with that understanding, let's move to section 4, the biomarkers that read the barriers state. And for that, I'll hand over to my colleague, Vaidehi.
Thank you, Komal, for that. Thank you, Komal, for that. Now, everyone has the basics of what is BBB and what is the journal structure of BBB, how it gets damaged because of some bacterial and viral load. Now, what is what happens after BBB breakage is there? Now, I'll take you towards the biomarkers and prognosis. First, let's know what is biomarkers. Biomarkers are basically some kind of indicators when something goes bad in our body or something uh, bad is going in our brain, at that time, some kind of indicators release themselves. If there is any kind of damage in BBB, some kind of small molecules start themselves to replicate in our body. Because of that, we can able to know that there is something going on. And these biomarkers, which are indicators, helps in prognosis. Now, what is prognosis? Prognosis is that which tells us that the infection at the later stage, at the future state, is it going to get worsened or it is going to get clinically downgradulated because of biomarkers. All this prognosis can be defined by biomarkers, and biomarkers and prognosis goes hand in hand. How that guys happens? Let me take you through that. Now here, when the BBB breaks, what first thing gets out of it? Because till the time the BBB is intact, we won't be able to get any kind of indications that there is a BBB breakage. But when there is BBB breakage, we will get some kind of indicators, biomarkers out of it. As per of the, let me take you through the Kashyap's 2016 paper, C3 Mouse 1. At that time, as Komal has indicated, at day 30, there was not a breakage of it. The barrier was intact. No biomarkers were released over there. Despite that, we can't say there is no infection in the thing.
Is there any issue with the slides?
Vaidehi, why don't you use Komal laptop? I think some kind of issue with your voice now also. Komal, can you hear me?
Yes, yes, sir. You're audible to me.
No, I'm talking, is Komal is there or?
Hello, sir. Am I audible to you?
Yeah.
Now Vaidehi is better. Go ahead.
Okay, thank you so much, sir.
You can share your slides.
Both terms goes hand.
The an issue.
Any issue, Komal or Vaidehi? I mean, you have to unmute yourself, Komal or Vaidehi. And presentation is also not there. Yeah, it's
Right.
there.
Yes.
Go ahead, Vaidehi.
Am I audible, sir, now?
Yeah,
Yes,
go
yes,
ahead, go ahead.
perfect. Yeah. Please.
Really sorry for that.
Yeah.
Biomarkers and prognosis. They're both the terms. Let me take you through again. Biomarkers basically are the indicators of the infection or anything bad is going in our body. And prognosis basically tells you the future of the disease. Will it get clear or it will get worse at the time with the time. So biomarkers and prognosis both go hand in hand. They are like indicator of each other. So, as Komal has told us, what is blood-brain barrier and how they get breached or damages, BBB. So, as per the like in this paper, as Kashyap's paper, at day 30, there is still the intact barrier, and no any kind of information is getting out of BBB that there is infection inside the BBB. All the environment in the BBB is silent till the time, but this doesn't mean that infection is not there. But the threshold of the infection is still low. That's why won't be able to detect anything at the day 30. Still, the despite detectable bacteria load is 4.0 log CFU. Colonies are still there, but we are not able to get any kind of indication from BBB that there is something fishy going in the BBB. So the brain, the barrier still holds that inflammatory response at its peak. It is not letting the barrier break and the antigens get out of the barrier, blood-brain barrier, and letting the immune system inside it. So till day 30, the threshold as it's low, we are not able to detect anything. But as the day passes and day 50 is there in this paper, so day 50 brain barrier is breached. There is damages in the blood-brain barrier. Now the threshold level of the infection has gone above that. The barrier has been broken. Now at this, after the barrier is broken, we are able to detect antigens. We are able to detect AG85P, RV2623 antigen. We are able to detect even immunological signals from our body like IgG against the antibodies, against the antigen of TB, and elevated ADA level. So all this science shows us that there is a BBB breakage at day 50 because the threshold is increased. As the biomarker signature of BBB breach requires both threshold bacterial load. It's not just there is infection or there is antigen or there is infection in your brain, but the threshold level should be that much high that the blood brain barrier gets break, and sufficient time also there, the bacteria needs the sufficient time that the bacteria replicate and damage the brain so that the immune cells get activated. So both threshold level of bacterial load and sufficient time for immune response to recurrent the cells over there, all the both the things are needed. So after that only the BBB break will be there. So BBB disruption is just not an immediate event. It is a progressive and dynamic one. It takes time. With the time, it gets break. After the threshold is done, the barrier will breach and the exchange of antigen and immunic cells between the BBB will be there, and we will dynamically be able to see the symptoms, signs of the blood-brain barrier breakage over there. Now, as per the in the paper of Kashyap sirs, we are able to see over here two graphs. For the first one, the antigens is on x-axis and ODs of it is on y-axis. In this stage, we are able to see antigen detection. This one is the controls of this mouse, and here we are able to see that CNS TB antigens are present. And this is the asterisk marks are denoting that the statistical significance that all the things or the cases we are able to see over here that the control the CNS TB's values are higher. The OD is higher than the control. Why? Because there is a real value, real observation we are able to see. It is not just any random kind of value we are getting, but the real values are getting. And over here, this is antigenic detection that there is antigen. And the second panel B, we are able to see that IgG detections we are against the antigen present in our body against AG85, 45 KD, ESAT6, all this, against all this antigen, there are some immunological antibodies present. Like the antibodies are trying to fight that antigen over there. So the BBB breach is there. So both these things tells us that the BBB breach converts a relatively silent infection into an immunological detectable inflammatory state. It's not just infection, it's not just immune cells, it's threshold and the time that immune cells gets to recover over there. So in this thing, we are able to see the antigen detection and IgG detection. Now, as of now, we have seen one biomarker, ADA, and other IgG detection. In the paper of Anitel, we are going to see the Q-albumin ratio. It is one of biomarkers, which is cubic albumin. As for now, as you know, albumin is mostly present in bloodstream. We won't be able to see in CSF or anywhere else. If we see there is something fishy or there is something kind of injury in your brain, in this paper, they have done CSF by serum albumin ratio. Means, is there albumin present in CSF by the ratio of serum, and it is an independent predictor of axonal injury. Over here, you can see this is your neuron, and there is an axonal. So if there is any kind of injury in your axon, so we will be getting NFL, which is a neurofilament light chain. When there is injury, this FNL will be getting detected in CSF and blood. So at that time, we are able to see that there is some kind of injury in your neuron or CNS injury is present. When even this is an independent predictor because in a neuroaseptic patient means the patient is having infection, but there is no symptoms. The patient is fine. This doesn't mean there is no infection, but inside we are seeing that there is exhaustion. NFL is present, not any kind of seizures or any other thing. We are able to see symptoms, but hidden we are getting NFL means there is seriously CNS injury present over there. In this paper, the evidence was like in 631 IVH infected adult, elevated albumin ratio was correlated with CSF. So in this, the neurotropin and leukocyte count is taken as an independent indicator. They are um, uh, inflammatory activator cytokines, which tells us that there is something over there, some bridge is over there. Then crucial finding is that the independent predictor, we can take NFL as an independent predictor in CNS injury. There is severe thing, we need to detect it and we need to find out what is wrong over here. Whenever there is a minor BBB impairment also, we like to measure the CNS damage. Little is equal to a major damage also. So this biomarker works as a various infectious etiology, and this biomarkers is very important because it gives us a hint before getting any kind of big damage before. There is going to a big damage, it tells us before time only. So biomarkers are important for us. Like now, let me get you through some kind of workflow is over here. Like biomarkers we get, but we need to take down a cutoff also. Like after what or after which stage we are able to say that yeah, this is a right value that there is an infection. So let me take you through like there is a table having a BBB state marker, published cutoff, and clinical correlation. When the BBB is intact, no any kind of harm is done to the BBB and no breakage is there. So we won't be able to see any biomarkers in our body or CSF, and no cutoff is also there. Means the patient is asymptomatic, no CNS infection is there. But when a little by little on a stage one, there is a bridge in TBM. How we'll be able to know that? First thing, we can go by CSF ATA. When there is a mild BBB damage also, now we will first be able to see albumin leakage in our CSF. By in this Kashyap 2006 paper, there is a 10 unit by liter, greater 10 unit by liter ADA is present in CSF, which tells us there is something neurological symptoms is present. Something is going over there, mild BBB damage is over there. With the clinical correlation, we can go with the neurological symptoms and MRI changes also for the addition of the symptoms with the ADA in the CSF. Then, as this legal stage, we can see the BBB is getting even more bridge. We can see albumin in our CSF, as albumin is mostly present in our bloodstream. But when it gets CSF, means there is some kind of axonal injury over there. In the paper of Aniston 2016, it is like 9 is to 10 to the power minus 3% of albumin is present in CSF, means more than mild BBB damages there, means neuroinflammation is also activated at that time. When a severe CNS injury is there, for that time or a viral breach or there is a breach because of a viral infection, we can see serum as 100 beta over there, which is a ligament B protein of BBB. So when in this paper of Green 2024, greater than 0.15 mg per ml of S100 beta is present in serum, which tells us that there is a very much severe CNS injury is present or aristocytic injury. As we know, astrocytic is a part of BBB, which is NVU, which is a neurovascular unit which makes the BBB unit, if there is in this endocytic injury is there, we will be able to see the CS100 beta in our serum. So as the stage goes on, like BBB is intact, but there is some kind of infection, we would be able to see albumin leakage. Severe in the second stage, if there is neuroinflammation, ADA elevation we will be seeing. Then when the CNS injury or a severe injury is present, at that time we can see NFL elevation over the serum or S100 beta over there. At this like this, we will be able to see that the something is going on as the stage gets increased over there. Till now we got the overview what happens when BB is breached, what gives a sign and what that sign means. But it's not like we have to know what the sign is and what is. We need to get some kind of therapies to stop all these things, and for that we need to know the BBB models, some kind of good models that will tell us that this medicine or this therapy is best, after which we can understand the BBB breakage, how it is done, and all. As we know, BBB is very intact junction. It don't allow any kind of thing to penetrate itself easily. In BBB, only small molecules, or at some time only, as Komal has indicated, that at some time only we are able to see that the medication is getting penetrated in our brain and it is able to clear out the infection, but not always. So drug delivery is one of the major problem because BBB is very much intact. So we have anti-TB drug. It's not that we don't have drug, but the problem is the efficient concentration of antibodies or drug is not able to reach over there, the site of infection. Especially as a meningeal inflammation is there, we won't be see that the penetration of antibodies are there. And there are evidences also in new ER et al. Paper that streptomycin, rifamycin are historical antibiotics which have a low CSF penetration. They don't get easily penetrate in our BBB, which is because of lipophilitic protein binding and efflux responder. Because all these three things make our BBB so strong, they won't allow the any kind of drug to penetrate easily at some kind of concentration. But first of all, over here, when there is infection is present, we give some drugs. But BBB won't allow that drugs to penetrate itself. Then inflammation, as the infection is increasing, there is damage to BBB. The inflammation helps that BBB to break and opens temporarily. So at that time, drugs enter betterly to clear it out. But as there is severe inflammation as a double H, so at that time it would, as cytokines also helps the infection to get it more and inflammations get more severe. So we start to give them steroids, and this steroids reduces inflammation, which helps the BBB to tighten itself again. It won't allow the drugs again to penetrate when the clearance is needed. At that time, BBB intacts itself again. So it is interpreted that we desperately need a better drug delivery system because BBB won't allow, and when there is inflammation, temporarily open ups, drugs get inside, but when steroids is also given to patient, the inflammation is there, reduces, and the tighten the BBB gets again tightened up. So we need a better drug delivery strategies. At that time, we can have a new strategies. We have a nanoparticles, we have intrathecal routes, and we need to focus on in vitro models, which will help us to know which drug is get giving us positive result, which kind of model or which kind of route we have to choose, at what time, what concentration we have to. So for that, we need to do clinical trials, and for that, we need to have a good models. And the key takeaway from this drug delivery problem is that the challenge is not just killing the pathogen, but successfully delivering the drug across the BBB at therapeutic concentration. We don't have to kill the pathogen. We have to successfully deliver that drug and don't allow that infection to get again over there. So selection of a model depends on a question you ask. What is your area? Why you need that? Like, if is there a new drug able to cross the BBB? Like we have your question, as no single model is perfect, but selection depends on entirely on therapeutic and mechanistic question you asked. And as we know, each model is different, and each model will give you a different answer because it depends on your research question, what you're asking on that. As of this, we have first question like, what does a new drug cross the BBB? So if we want to know that a new drug will penetrate BBB or not, we need to have a high throughput query. We have to run the test, we have to run the model in a large amount. For that, we have a best model which has a organoplate, have a high throughput one. Here, a 40 plate chip plate is taken, and here, cell culture type on chip, a tissue or a cell is grown on a chip over here on a microfluidic. On this thing, one tissue is grown, and we are able to work on that one tissue. What happens to that tissue when we give some kind of drug or some kind of infection is given to that thing? So at once we will get a enough in number of the results out of it. And here also, why this organoplate works, because it gives a tear readout, means the tightness of the BBB is there or not. With this tear mechanism, we able to do that. Then second, if question is like, does a mutant strain cause a neuroinfection? Like C3 mouse model of Kashyap says, like in this also we were seeing like there is dissemination, invasion of bacteria or not. So this was in this model. Why this model is good? Because it was a whole organism. Like first one, we are not just working on one tissue, but we are working on a whole organism, which will tell us the interactions and which will give our longitudinal survival data. As we want, we will be getting the data, but it is not a human model. There is some kind of difference in human and mouse thing. Some difference will be there. So this was not there. So this we will depends on the C3 model, the whole organism. You are able to see that there is a dissemination, there is infection from lungs to brain, like that. Then the third question is, what is a molecular mechanism? If we are asking that any kind of molecular mechanism of breach is over there or not, for that the best model is iPSC derived, which is induced pluripotent stem cells. As we know, we can turn a pluripotent cell into a specific cell. So if we want to know what is the genetic and cellular interaction happens between the cell, so we would give a one indicator to it. So it can contact and make a new cell which is there. So in NVU, like we know, neurovascular unit models, erythrocytic parasites, endothelial cells are present, and all this make a unit of BBB. So with this induced pluripotent stem cell, we will be able to know which kind of genetic and interaction happens. In this paper of Galileo 2025, they have done this only to know the interaction, how one cell gives a generation to a new cell, and what is the interaction happens over there. If a question is, how can we restore a barrier functions? As we know that best model for this is parasite co-culture chip. This is also an cell culture one. Here also tissue is grown, but in this co-culture chip, we can grow a different tissue at a one chip also. Over here, we can grow two tissues and simultaneously work on it. So what happens in a particular tissue when different concentrations or any kind of it's given to drugs are given to them, what kind of interaction happens, we'll be able to see over here. As parasitic is also a part of NVU BBB, it's important to know. So this test or this model works because this test specifically for therapeutic candidates, for knowing the tight junction and loose junction, what happens when a different kinds of drugs or inhibitors are given to them. This for all the models we can do, and it depends on the question we ask or what is our output we want. At that time, we go to models, as all models are not same, because BBB is also a versatile thing. So one model won't be able to give us our answer, but it will give our way to the another question or another answer or another model, which will help us to know better the BBB. Now we have known about what is the problem, why there is no reach of a antibodies to the BBB or center where we need. So we need to think about some kind of new ways to provide that antibodies or things to our BBB. So first of all, we have a host-directed one. Over here, what happens? We are going to use the immune response generated by our own immune. Over here, this MIF is a microphage mediator inflammatory factor. In this is the thing which reduces the blood in the JVE. It reduces the blood weight. Why? What happens? MIF is a when there is an infection. MIF is an inhibitor. It gets produced in our body in n number. So there is inflammation in our brain. What happened in this if we give iOS 1? It is an inhibitor. So this thing goes and reduces that inflammation in our brain, which is MIF, and we can use those things to inhibit that immune system to our body and is the viral load and neuroinflammation. However, it just gives us 20% mortality reduction, not more than that, but this tells us that we need a combination of different therapies with it. Then second was a strain specific target. As over here, KNFT. KNFT is a protein which helps MTB to invade in our system, in our BBB. It gets attached to BBB and protein, this protein of TBM helps to invade it into the BBB and give an infection to us. But if we attenuate that PKND and attenuate that bacteria and disseminate it into our brain, it will reduce because there is no thing that attaches to the RBBB and helps it to invade it. So against only, so we are attenuating the PKND. There is no protein to of the bacteria. So how will it would engage in Guinea pig model? It gives us the results that these things can be used. This model can be used. Then drug delivery with the help of nanoparticles. As we know, BBB is a tight junction. It won't allow anything to invade itself, but small particles can invade it in the BBB. So for that, we can take help of nanoparticles, which is a carrier, which can carry a small concentration of amount of antibodies and travel it to inside the BBB and can give a treatment to it. So with these nanoparticles, we will be providing a slow and a stable amount concentrations of drug to the things, and it will help to recover or clean that infection present in our brain. So these were the three therapeutic actions which we can use in nowadays or in a future or in coming future. Then we need to criticize the limitations of all the models or we have discussed till now. And the mouse model, it helps us to know that there is a dissemination of infection from lungs to the brain, but no BBP integrity markers were measured or aerosol route was not validated at that time. So at that thing, but that tells us that there is a dissemination. In Nair et al., it was in vitro chip model, but the model lacked the endothelial monoculture. It was not able to tell us that there is kind of infection, and it lacks vital erythrocytes and pericyte cells, which makes a building block of BBB. In Galileo paper, the 3D iPSC one, there was a lacking of microglia and neurons, which is an important part of BBB, which keeps the BBB intact. And shear stress was also not fully studied over there. The fourth model which we have talked about was the Greens et al. Model. In over here, the small sample size was taken and it was not done on large sample size, so we won't be able to tell this is the thing or this was the cutoff, this was the result. So we need a large amount of size of samples that have to be done, and it also lacks the direct CSF biomarker correlations over there. So there is no one particular model perfect or does exist. The field must need to do or cross murine or human chip and clinical core data. We need to look over for a new things that which will tell us about more about BBB and how we can restore the BBB from the infection. The key takes away from all the sections that we have given, like from section 1 to section 5, that we know that BBB is a very dynamic barrier. It won't allow any kind of things to pass it on. It is a selective memory. It is a very good memory which protects us from all the pathogen all the time. Then second is that strain matter. It's not like that any kind of strain would be able to harm BBB. So threshold is important, the time is important, and the strain which is infecting the BBB, that is the main thing, which is sufficient. If it's insufficient, the BBB will not break. The friendly fire is like that neuroinflammation helps us to recruit the new immune cells, but sometimes it works as a double-edged sword. It can worsens the response or worsens the disease over there. So we need to look out over the neuroinflammation at the time where we can stop it. If it is in helping mode, at that time only we need to do something so that injury won't get more worsen. Then we have a biomarkers readouts. As we have seen that biomarkers tells us after that BBB is bridged, then we need to have a protection of the BBB. So biomarkers is the one which helps us to denote the prognosis of the disease also at later stage, or we need which kind of therapy is needed over there. Then just it's not just simple to know that BBB is breaked and there is infection, but we need to know that we need a models that which will help us to uh, get a knowledge of which drug, which route is important for us to treat that BBB breakage. So we have the tools, the biomarkers, and the mechanistic understanding, but the remaining gap is translating this into clinical practice. But for the first time, the gap is measurable and actionable. First, we need to know there is a gap. We need to know, we have to have that sense that we are in urgent need to have a new model to understand this BBB and what issues are over there and how we can resolve it. Thank you so much for your patience, and the floor is open for discussion. Thank you.
Thank you, Ms. Komal and Vaidehi, for a very comprehensive presentation. I think that was quite an enlightening talk about the complexity of the blood-brain barrier. And I think we have deciphered maybe a part of it. So much more is yet to be understood. Uh, um, Sir can give his comments on the
Yeah.
presentation.
Yeah. Thank you, Angel. And thank you, both Komal and Vaidehi. Actually, it's a thought-provoking discussion, basically. The talk is like that. Basically, it's coming, a lot of questions are coming out. Even I've worked a lot on these things, but when you are presenting, even I am getting a lot of questions on these things. And we understand, actually, you have highlighted that the PBB is not only a kind of a static anatomical wall, but it is highly dynamic. And what we have understand, it is very immunologically regulated interface, basically. So that's what, if we talk about, it influences endothelial cells, astrocytes, microglial cells, immune mediators. So there are so many things are there which are going to be involved in these things. And one of the most important take-home message is that that neuro invasion is not only determined by pathogen alone. That's what the experience we have with previous paper and the paper these people have presented. Basically, it's an interaction between pathogen, then host specific genes are there, inflammatory pathways are there, and which they haven't discussed. I think the one more area which they haven't touched is gut. So basically, when there is an inflammation in the gut also, they release a lot of pro or anti-inflammatory cytokines, and basically they go to the blood-brain barrier and damage. So we haven't touched that gut-brain axis angle into that, but that's also playing. So that's why gut and brain are always linked together with all these things. So this is basically decides whether BBB remains intact or it is going to be breakdown basically. And we also saw some of the genes which they have reported, claudine and other genes, genetic factors, regulatory, do inflammatory signaling and all these things. So this open, like what in the end they have indicated opportunity for translational research. We need to figure it out, biomarker discovery, host-directed therapies, and I mean, not yet, but I think ultimately, I mean, all the people, those who are here, I feel someday we have to go to a host-specific therapies. Actually, that is what we do at the bedside, right? Not every medicine on the book style work to everyone, right? So we need to change, we need to modulate. And the best thing which I have understand, I'm trying to work on it, is the circadian rhythm of that BBB. If we can work on that with some kind of a MRI label, some kind of a dyes or some kind of a thing, if we can start figuring out whether we are getting some kind of a BBB report also in the MRI, some label, then it will be useful to schedule the treatment and all these things. I mean, this is all kind of a things which coming from research-based things. So it's a very complex topic. We all understand it, but you have present little in a structured manner and raised a lot of questions. So thank you once again, and maybe Angel, you are going to take questions.
Yes, there are
Or
two questions. I
yeah.
will open it for discussion. How do host-specific immunogenetics regulate the inflammatory pathways that lead to blood-brain barrier breakdown and subsequent CNS invasion by pathogens? And are the presence of some genes influence the host susceptibility to more severe outcomes in neuroinfection? So I think do they want to take the questions, or would you?
Komal or Vaidehi, are you there? They have gone. No problem. The time they'll join. I mean, I'm coming to the question of answer of that particular thing. If they'll join, I'll ask them to repeat again. So basically, I think I have already indicated the involvement of endothelial cells, astrocytes, and all these things are already there. So I think they are the things which actually influence the BBB is one of the things which we think it involves for breakdown. And some specific genes, yes, they have reported around 5 to 6 genes. I think if you have gone through the presentation, they have reported. And we have sent samples to one lab where they have indicated, I mean, there are some, we have done microarrays. We haven't shown that data. When the pathogen really crosses the blood-brain barrier, there are some group of genes overexpressed during that time. And when it is not, the genes are different. So signatures of biomarkers is definitely going to play some. Vaidehi, are you there? Vaidehi or Komal?
Yes, sir.
So, I mean, it's better if you can answer second question.
So the second question is, since ADA is considered a relatively non-specific inflammatory marker, is there a strong evidence from diagnostic accuracy studies or meta-analysis supporting its specificity for CNS TB? And how much weight can we place on ADA elevation alone? I think this is one thing which we are always perplexed with because though even pleural fluid ADA, CSF ADA, we regularly send it, but it's sometimes when the gene expert comes negative, can you call it TB just based on ADA levels? Because the specificity of it's just a marker of a blood-brain barrier breach. So how do we take the specificity of it is the question. So any answers?
Uh, There was a meta-analysis study published in the Singapore Medical Journal, which exclusively states that if the ADA levels are greater than 10 liter per the value which IU per liter, which we already discussed in the presentation, it is an exclusively shows that the patient is TBM positive. I think the ADA is not a standalone confirmation, but when combined with the clinical features, it could be a great a diagnostic panel test.
So the problem is, we know CNS TB is a paucibacillary disease. So most often we will not get a microbiological confirmation. So many centers actually, many places they advocate, ADA, just one value, above the cutoff. You can label that as TBM. So maybe that I would take it with a pinch of salt, sir.
Yeah,
Rajpal.
I just want to tell you, I mean, let me tell you very honestly, ADA is not a marker for CNS TB, right?
Yes.
No, no, it's not a marker. And not, we have shown in this study also, they haven't shown it's a marker for CNS TB. See, once the TB is diagnosed, right? One the brain TB is diagnosed, if you get a follow-up samples or do experimental study, longitudinal kind of a thing, the expression of this T cell activity will be useful to tell.
Oh.
We cannot use it for diagnosis.
Diagnosis,
Yeah,
yes. That's what
even we
that
have
was
published in BMC, cerebrospinal fluid BMC journal, where we have reported ADA in a different way. And here also, these people are not saying that it is a T. When ADA's expression is high, that means it's a TB, right?
Okay.
So they're saying it's a TB, and then ADA expression is high. Then it makes sense with the follow-up samples and all these things. Whether it goes down, whether it goes elevated, then it has some kind of some kind of prognostic marker with T cell activity and all these things.
Yes. So other comment I wanted to ask was, there are a lot of upcoming markers like this GFAP, neurofilament light, especially in the context of immune disorders, MS, um, neuromyelitis optica spectrum disorders. There is a lot of output on this. And I think in the COVID era, they had looked at like elevated GFAP and neurofilament light as a prognostic marker. And also there was a, if I am right, there was a recent publication on its implications. If it is elevated when you have COVID, what is the chance of them having a long COVID? That is all those long COVID symptoms and other things. So I think that's also worth exploring because, but the problem again here is GFAP and NFL goes up in any insult. There are studies on traumatic brain injury, there are studies on stroke, epilepsy, infection, inflammation, everything. So again, to find a specific marker is the issue. So, what is your opinion on that, sir? On
No,
these new
actually, that's
markers?
what I'm saying, because here in the BBB, we are not working on the diagnostic angle.
Yes,
So
yes.
we don't need any kind of because already established marker and all these things to prove that the BB is breached
Breached.
or not. That's the thing we are working on this area. But if I take it as a diagnostic angle,
Yeah.
definitely there are so many non-specific things are there, which like ADA, they are useful. Sometimes it's useful. Like we work in stroke. I'm going a little bit to brain TB. We
Oh,
work on the stroke, and the BBB is useful in that case because when you have a glial cell damage, neural cell damage, then NAC and GFP, all these things we are getting in the serum also,
yeah.
right? So it's a reverse kind of the advantage and disadvantage basically. So there's not a lot of specific markers, and we all know, Dr. Angel, when the patients come, sometimes something works, sometimes don't work.
Yes.
PCR is positive, antibodies are not there. So this we have to, at least I accept it honestly, if we don't
Yes,
understand
yes.
it much with that thing.
Yes.
So signatures are signatures. It's like traffic, right? Sometimes they move in that way, sometimes they move in different way. But definitely they are signatures. So if we don't get anything, we have to take support of that. And ultimately, like what I've said in my presentation a couple of weeks back, we have to decide it. Basically, how we have to go it.
Yes.
Yeah, I think it's interesting to see in the brain TB. Just I'm thinking whether people have seen this this S100 beta beta and NSC in TBM also. They have
Okay.
seen some kind of a damage and seen in serum also this kind of this thing. So, but it all depends on patient to patient. But I think cytokine is the thing which neuroinflammation angle. If you look into that, this is what we are doing at the moment. In-depth study with follow-up things and follow-up patients, how they behave is very important. It's a kind of a thing which might be useful in future also.
Okay. Sure. I think there are no further questions. So I think we can, that was very nice, comprehensive presentation. So keep up the good work. I think you're literally multitasking, and I'm happy that you're able to do this. Thank you, sir, for giving them this opportunity and sharing your experience in this journal club. We'll be soon sharing this presentation in the YouTube channel also, so that it will be more widely seen. Thank you.
Right. Thank you so much. Thank you so much for giving this opportunity.
Thank you,
Thank you,
ma'am.
ma'am.
Thank you.